So our next speaker is Miguel Lopez Ramirez of UCSD. He is the Assistant Adjunct Professor of Medicine. His research has been focused on studying the molecular, genetic, and cellular mechanism that regulates the vasculature in health and disease.
Thank you. Well, thank you everyone. It has been a very exciting meeting. I get to know a few of you, and it has been a real pleasure, actually. So I'm going to explain a little bit. I did I started as a scientist when I was, I guess, 7 years old. I was always motivated to learn about diseases. And actually, I had the opportunity to do my PhD in England, where I learned about how the vasculature forms, how the vasculature helps us to be healthy. And I was also had the opportunity to be with a very great mentors that actually teach me about a lot of immunology. And we were always inspired not on publishing and publishing, rather than learning well and do the task slow but good, steady. And actually, it's what I'm trying to now pass it on to my own students. And literally, since I was very interested in the understanding the blood vessels of the brain, I got very interested in studying CCMs because it's a condition that a single gene could alter the program or the function of these blood vessels very specifically in a very less extent other blood vessels in our body. So that's really intrigued me, and actually I was at Yale University doing my postdoc, and I met my mentor at UCSD, Mark Ginsberg, who was studying this disease, and I started talking to him, and he said, well, why don't you come to UCSD to learn more about that? So that's how I moved. But my mentor was studying communication between cells and cells, or addition of the blood vessels, but my main interest was why the CCMs affects primarily the central nervous system, and why these vascular malformations can be quiescent, and what makes them becomes worse or activated. That's what I'm going to be referring, like quiescent, like silence, and activated, like when they give manifestation of the disease. So my main interest is to understand these drivers that can do from being here to be here, and I started looking at environmental, genetic, and biological factors. Many of these questions came inspired for having walks at Angioma Alliance with Lindsay and Linda, that we actually got inspired, and I want to talk to you a little bit of the stories that we learned by talking with patients, talking with the people who is affected, that we hope we can help with our research. So, So the way that we study in my lab is basically we do an integration of multiple models and also working with multiple people in order to all understand this disease at different levels. And here is a little bit of the schematic way that we do this. So basically, we can do tissue culture from animals that can develop the disease, and I'm going to explain to you advantage and disadvantage of how we make these models and why I think they are very important. But also these cells that are coming from the animals can also come from patients, actually, that we can also, when you have the surgeries, we can have access to that material. And in that way, what we have done is that we have been also working with a company that is called Benevolent Artificial Intelligence. So what we have done is integrated all the knowledge that we got in tissue culture from the animals, from the experiments in cultures, but also with bioinformatics that is out there in the web. And all this knowledge goes into a computer and literally give us potential targets that we have been assessing over the time. It's going slow, but it's moving, I guess. Okay. So why is it important to generate animal models? Or models in general? So as you can imagine, obviously, when we have a model, we can make small changes and see if those small changes have benefits or has repercussions. And unfortunately, I will not say that I'm very proud of using animals, because I consider them very, very important, but is one a very strong model to understand the evolution of the disease and to try to understand what can make them better and prevent the disease. But this model, literally what it works is I call it Marchak or Matthews model, and I'm not lying because I just mentioned it in the other audience, but Marchak is Doc. He has been the discoverer of the CCM1, CCM2, and he was telling me that almost CCM3, but he didn't. But actually, he has done a lot for the field, and he's a very, very good friend as well. So he shared these animal models, and we have been studying them because it recapitulates several of the characteristics of this condition. And we can also look at different stages, and as you will see, I mean, I do a lot of research in particularly, I'm very interested in pediatric cases, and I will explain why. But this model, literally what it consisted is in the case of CCM condition, you have a loss of the function of the gene. In this case, what we do is that we genetically inactivate the gene at very specific time points. So we can decide when and where to inactivate the gene so we can study that process that makes the changes in the vasculature over the time. And in these models, let me see, okay. So here is very important. This is what we always evaluate to consider a good model, because when we call it model, it's like something that we can get information that can, in certain extent, recapitulate what we wanted to study. And in a model, as you can see, it's not perfect. But as long as we know what are the points that we can take from that model, it makes our analysis very strong. Because we don't use only one single model. As you can see, we have been over the time, we have been changes our model so we can make it always more close to what the condition is. And in this case, you can see here some of the characteristics of the CCM in the vasculature and throughout the brain. I'm not going to go one by one. And here, that's why it says yes, 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 yes. And as you can see, independent of the model, some of those characteristics are present. But as you can see, it's like having an iPhone. You know, you go from iPhone 4, and now we are in iPhone, I don't know if it's 14, 15, but this is the iPhone that we are getting over the time. We try to adapt to the best conditions. So right now, we have basically prepared a new model that develops only single lesions so we can monitor over the time one single lesion how it evolves over the time. And I will be very happy to go into more detail, but I know that the time is also, time is important. But overall, what I'm trying to say is that we always try to get all these models to recapitulate at the more extent the human disease. Using these models, we have done good contributions to the community. I don't know if it's in the next slide. Oh, okay, maybe I didn't put it, but I can go here. So one of the contributions that we did very early when I was a postdoc is that we understood that this vasculature was hypersensitive to growth. In other words, they like to grow. And we explained the mechanism of why they grow so fast. So then, during the time when I got like a transition to become independent, I understood why I was very interested to understand why these vascular malformations bleed. And then we understood the mechanisms of why they cause the bleedings. And that is very important because it gives us an understanding that what are the therapeutics that we need for growth, for bleedings. And I will explain a little bit why it's so important knowing the mechanisms, the basis, because many people will say, well, let's just try drugs and let's see which one blocks the disease. That is a very good approach. But unfortunately, sometimes it cannot tell us the whole information of why it does the effect. And knowing what it does the effect, it may be personalized. In other words, some patients may need one type of drug while other patients need another type of drug. So the basic research is based on that, on understanding very particularly how the disease evolved, what are the important points to block, okay? So one way that we do it is that we can imagine that you have the brain in these animal models, and then we can dissociate in individual units. So we can dissociate the brain and get only the vessels, the endothelial cells, the ones who are like our veins in our brain, and study them to do a high-profile analysis of genes. We can also look at other cells that are part of the residence brain cells and look at the profiles. So the ones that we saw that have like the major changes during the disease were two types, actually, the endothelium and the astrocytes. Then here are a little bit of how we visualize those changes. But what we learned, then what we could introduce to the field, is that the vasculature by itself does not cause the disease. It starts the disease, but it's not sufficient to produce the whole disease. It needs some kind of feeders. And that makes sense a little bit because the endothelium wants to grow, but someone needs to provide them with the food or with the source of energy so they can grow. So we have been identifying these mechanisms of how the blood vessels basically form these abnormalities. And more recently, working with Dr. Helen Kim and Chantel Weitzmer, they have been doing a lot of analysis from patients, from blood cells, and looking at polymorphisms. Polymorphisms are like changes in the DNA that do not transduce like a completely loss of function of the gene. It's just some small modifications that may or may not influence a disease. So by doing that, I'm not going to go into more details, but what you have is that you have two different communities or groups. One that is, let's say, that is this DNA that is very common, is very, very common. Millions of people has that. But at least 5%, they may have a change. And here you can see this is what we read the DNA, like ATGs, which is very complex. Looks very simple, but when you have a whole sequence, it becomes very complex. But as you can see, let's say 10 million of people have this sequence on the top, but only 5% of that 10 million, they will have a small change here, but do not transduce in any big change in the condition of the patient. But well, what we did is that we actually used, I will see if we have a little bit of a different slide. What we did is basically we cross-species interactions. We see, okay, we have this information from the animal models. Let's see what is in that information that you have with human patients. And that was very important because literally, it's here. Literally what we are doing is what we learn from the animals, what could be or not be in the human populations affected with this condition. And actually, we identify one particular gene that I would like to talk to you a little bit because we consider that it could be very important in that transition that I was calling you between being quiescent and being active or aggressive. And this gene is called CX3CR1, which is a chemokine receptor. And this chemokine is like is a way to communicate between cells. If you remember, I have these cells that were communicating between the endothelium and the astrocytes. But what we learned from this one is that it's more complex than that. So basically, these chemokines allow you not only to communicate between the endothelium and the astrocytes, which now we know it does, but also communicate with the immune system. And towards that, we have been understanding how the immune system actually contributes to the disease. And now to make it more complex, but is again complex, but trying to visualize in a simple way is very good, is that the immune system does the two. It does the good things and the bad things. So right now we try to understand how to isolate the bad things and leave only the good things so we can make the lesions always remain quiescent. And I have 3 minutes, so I will speed it up a little bit. This is my last presentation. I'm happy to discuss a little bit of how the environmental factors, how the biological factors we are studying, we are contributing to the knowledge of this disease. But this is something that we are very, very interested in. Actually, we are working on that, very important. As I mentioned, the CX3CR and CX3CL1, which is the way that they communicate, they involve the immune system. So we are doing modifications in animals where we can alter this communication and see how the the vascular malformation or the abnormality, these lesions evolve. And we are using, or we are collaborating with the clinicians to provide us with human specimens, particularly in the pediatric stage, because we think that that could be a better process to understand early stages and understand before they become too chronicize. So I think that will help us to better understand the signatures that can drive between being quiescent into a more active lesions. And I think my last slide is this one. I think the list here is actually short. Now I have more students. They are the ones who has contributed enormously to this work. Some of them, most of them, actually very proud. They have gone into med school. Here is just a small list that they have been contributing in one of the presentations that I actually copy-paste, but they literally, Catherine did a lot of the work for the inflammation. Angela did a lot of the initial work for the hypoxia, and currently Edu and Elios, both postdocs, they have been doing the studies that I show you today. So thank you very much, and I'm happy to, if you have questions, happy to answer them.
Anybody have any questions?
I missed one of your slides, but you were talking, this is all around the genetic components of CCM, correct?
Yes,
So
mainly biological and yes, in genetics.
So there's not the sporadic, say, study of some of the neuroinflammation that happens in that process, correct, at this time?
No, it's very interesting. Should I tell? No, I will tell you. Yeah, so the lesions, even if it's sporadic or not sporadic, they may behave biologically identical.
So genetic malformations.
Because they are the same drivers.
Okay.
So right now we are developing new models where they can have only single lesions that will resemble a little bit the sporadic form, and they also become inflamed.
But the mechanisms behind the genetic change is still the same.
No, that's the good news. The good news, even that it's a very complex disease, once they turn aggressive, in our models are the same mechanisms. It's not only one. That is the only thing. It's not only one, but they share similar mechanisms. And in this case, even when we are looking mainly CCM1 and CCM3, again, the good news is that it's similar mechanisms. So that is actually very motivating for us because we can potentially develop therapies for that.
So you mentioned that there were some of the triggers and environmental factors there, and I don't know how much time we have for that. Are there any maybe highlights of that you can share with us?
Yeah, so I always, this is an important one. I think in the Angioma Alliance in the website is there, so I guess I can mention it. But for several years, we have been working with one particular environmental factor, such as hypoxia, which is low levels of oxygen, and at least in our models, is a strong driver. However, the mechanisms that our rodents develop are different than the humans, so we are still collaborating with clinicians to understand at what extent these particular models that gets very enhanced with low levels of oxygen could contribute also to the human disease. Because it's very important, again, there are models, and as I show you, not all the yes matches to the human disease, so it's very important for us to, before we go to the whole community, we need to know what aspects of the hypoxia could be a driver, like such as sleep apnea, high altitudes, extensive exercise, anemia. Those are factors that potentially could contribute to increase the disease in our models, is what they predict.